In this demonstration, I'm going to run a complete RNA-seq demonstration, and I'll start off by going into a data library and selecting some pre-prepared data, which is very small. It's only got 2,500 reads in each file, and I've just imported that into the history that I was working on when I began. I'll rename this RNA demo so I can find it again. Anytime in the future that I come back, I can select the RNA demo history. And at the moment it's got one file in it which has a mere 2,500 sequences, not the 25 or 30 million you might expect, and it's in the usual FASTQ format. So we always start out by grooming FASTQ data when it comes into Galaxy to make sure that it's in the correct format, and this has come off the Baker sequences, so it's Illumina 1.3 data. Click execute to get that running, and I'm going to run a quality control test on the data also by using the FastQC tool from Babraham Bioinformatics. I'll just click execute, accept all the default options, and those two jobs have now started running. I don't really need to wait for them. This one's now executing, and the other one's still waiting to get in the queue, but I can still go to mapping and select map with BWA for Illumina, because that's the kind of data I have, and I'll use a built-in index. I'll use HG19, it's a bit more up to date, and I'll use the groomed file that's ready to roll, except the most common BWA settings, and away we go. And there's one more thing I need to do, because the BWA program produces a SAM file, and my next step requires BAM for efficiency. So we choose SAM to BAM, select first on, that's this file. Yes, alright, so everything's ready to go. Let's have a quick look at the FastQC output. Remember, we always run QC because we like to see green ticks, because that means FastQC thought, for example, the basic statistics were looking good, which they are. You can see that you've got very relatively good preservation of the quality scores across the entire range of length. It drops off as you get to the end. FastQC is really, really cool, and has lots of very interesting reports which you should look at very carefully, particularly the overrepresented sequences. If you've forgotten to clip adapters, you'll find them there, or this KMA report, which will tell you a lot about adapters you forgot to ligate. But this has only got 2,500 reads, so it doesn't really have a lot in it, and I don't want to spend a lot of time going over it, although you can learn a lot about your sequence. The other steps are complete, and BWA has finished running, and it produced a SAM file. You can read all about the SAM format at the SAM tools site, but essentially it has a header which tells you what the reference sequence is, and then it's got, for each read that was in this this FastQ file that came in, you'll find the read now has a mapping generally to a chromosome and an offset. And for example, this read here didn't map anywhere. This one mapped to chromosome 1 at this offset, and there's all sorts of valuable information here in the SAM file. But for our next step, we actually need a BAM file, which is a binary file. We can't look at it directly, but it's essentially got all that's in this file, but it's much smaller. It's only 136K, whereas this would be probably 5 to 10 times as big. Alright, so that's all very good. We have run a groomer, we've run FastQC, we have a lovely quality report, we've mapped the data, and we have a SAM file, and now we have a BAM file from all of this, and that's very good. But one thing that we're going to have to do is run this same set of activities over a lot more files, because this is part of an experiment where there were multiple lanes of sequencing and many samples to be compared a little further on. So I'm going to use the extract workflow option from the options that are available in my history, and I want an input data set, I want a groom, I want FastQC, I want BWA, and then I want SAM converted into BAM, so that's all good. I'm going to give it a new name. We'll call it RNA demo, and click create workflow, and Galaxy has just worked a little miracle, because if I go up here into my workflows, the first one that you'll see is this RNA demo. Let's edit it. We get a graphical user interface where we can relay it out so it looks a bit neater. We'll just get rid of that. And here we have the input data set going into the FastQ groomer. I'm going to click the little snowflake so the file's persistent. One of the outputs from the groomer, or a copy of the output, goes to FastQC, which does the report. Another copy goes into map with BWA for Illumina, and then the final step is the SAM to BAM conversion. So I'm saving all of those outputs. I'll click Save, and we're good to go now, so we can close this, and we can go back to our original history and click Workflows again. Just want to check where I was. Choose RNA Demo, and this time we're going to run it. And actually, we forgot to do something, so forgive me. What I meant to do is go to the data library, go back to the demonstration sample RNA data, and I've already got this one. I actually need this one for the next step, and I need all of the rest because I want... Actually, I'm only going to choose the HG samples and the control samples, so I'm only going to do another 5 samples, plus the exons that I need for the next step. The default is import to current history. Click Go. Galaxy gives me references to all of these data sets, so I don't actually get copies. But it looks like I've got copies because they're now in my history. So back to Workflow, and choose the one I just made, choose Run, and this time I can set this up properly. So I want to run the workflow over everything except data 1 that's already done. So I want all 5 of the available input files, and I basically want all of these steps run, please. So I click Run Workflow, and Galaxy is now going to run the same steps that we ran earlier that you saw me do by hand, but it's going to do them all automatically, one after the other, for each of these input files. Now, you'll notice there's a bit of a sort of nervous pause. There we are, Galaxy's come back. This is running inside the baker. It's only a little SunGrid engine with 2 machines connected, one with 16 cores and one with 12, so it's not a huge machine. But now you can see my history has all of these running jobs, which are all going to end up producing FastQ, well, Groom, then FastQC, then align it with BWA to produce a SAM file, and then convert that SAM file into a BAM file. And every time a job finishes, it goes green. You can see that this is proceeding very quickly. Of course, that's because I've got only a tiny number of sequences in each of the input files. If we had the original input files before I cut them down, they'd each have about 25 to 30 million reads, and this whole procedure would take about 1,000 times longer, so it would run for a couple of hours. However, we're nearly there, which is very good. The very last step is now just about finished, and so here we go. We've got a BAM file, a SAM file for this input, which is one of the HG samples. We've got the FastQC report, which you've seen before, made up just from a sample of the groomed and the original data that I'd imported from the library. So we've now 6 of these final mapped files that we need. So now we go into the Baker IDI testing toolkit, and you'll see there's lots of choices. One of them is multiple BAMs to matrix for DGE. That's the next step in a RNA analysis. And we're going to add this BAM file, but we're also going to add a whole lot more. We're going to add 5 more. This is very difficult to do cleanly, so I'm going to do it. That's 2, 3, I'm going to 4, so I'm going to do 5 of these, and then I'm going to go back and point them all in the right place, because the first file is 31, so I don't want that twice. What I want is number 27 next, and then here I want number 23, here I want number 19, and here I want number 15, and last of all I want number 5. So these are all going to be merged together into a count matrix that we're going to be able to run EdgeR on. I'll leave all of the defaults, and I'll just click execute, and this step is one of the longest ones. It does actually take a while to run. It takes about probably about 5 or 6 minutes. So what I might do is just put in a little pause here. This is a point at which we'll come back once the file's prepared. So I'll see you in a moment. Okay, we're back, and this is finished, and it's produced a nice matrix. which is ready for the edgeR program in R. And the matrix looks like the names of each of the files, and in each column is a count of the number of occurrences in each file, in each BAM file, that is each sample. And that has been counted using this file, this HG19 exons merged gene name.bed, which is a bed format file, chromosome start end, and it contains every exon of every gene in RefSeq. So the first few genes are all WASH-P7, and they're going to end up in this count matrix as one single row for WASH-W-P7, or whatever the gene name is. And in each row there's a series of counts. And these counts are very small because I only ran this amalgamation program, this BAMS2MX program, over 6 times 2,500 reads. So there's not really much data there, but I just wanted to be able to run it reasonably quickly. So for digital differential expression, we're going to use this file now, number 32. And for treatment, we're going to select the samples which have control in their names. And then for, we'll do that the other way around. For treatment, we're going to select the ones that have HG in their names. Let's just get this right. And for control, let's change this to the name HG just to remind us. For control, we're going to select the control samples, so the ones that have control in their name, these two and this one. And then we're going to use edgeR, click execute, and Galaxy will now run edgeR over the counts, the digital gene expression counts in each of the samples, and I've set the experiment up so that edgeR will look to see if there is significant differential expression for each gene across the two different kinds of replicates. And edgeR is the appropriate method to use for RNA-seq data because, well, it's pretty quick, it's finished. edgeR gives two outputs, one of which is this HTML file, and that gives us a multiple dimensional scaling plot, which shows us that the controls and the controls are sort of a bit more over the place on dimension 2, but they're all pretty much lined up on dimension 1, and the cases, in this case the HG samples, are a little more clustered on dimension 2, but perhaps a bit more spread out on dimension 1. So in other words, this is a pretty unsatisfactory plot for lots of reasons, but of course remember it's based on a tiny sample of the worst reads, the first 2,500 reads. Normally with millions of reads you get a nice dense smear plot. There's not much to see in this rather silly example, except that none of them were significant by edgeR, which shouldn't surprise anybody because there's so few reads. But this HTML file gives you the R code that was run. If you want to run it again, you can just click there and get the R code and run it under the R interpreter. And the final output is this this differential gene expression chart, a kind of top table, which is, sorry you can't see it very well, there we go, which shows that this is the top gene, this is the log concentration. You need to read the edgeR documentation to understand what these things mean. Log fold change, you can probably guess. P-value unadjusted and then adjusted for all of the tests that we did, and of course the adjusted p-values are not significant, which shouldn't surprise you because we have almost no power with only 2,500 reads. But at least we do have the correct statistical method applied to digital differential expression, and it's all done literally with a few mouse clicks and a little bit of patience using the tools that are currently available in Galaxy. So I hope you enjoyed the demonstration. I hope you understood what I did. Let me just remind you, we started out by importing one file from a library, and then we ran the groomer over it. We ran FastQC to get some quality reports, and then we ran the SAM tools, the BWA mapper, and that gave us a SAM file. We converted that to BAM. Then we extracted a workflow by going here to Options, Extract Workflow, and then we saved that workflow. We edited it a little bit to clean it up, and then we ran it over the other 5 files, and then we ran this this program here, the one that does multiple BAM files, multiple sets of reads to a count matrix for DGE, and then we ran the DGE tool here, and that took us a little less than 15 minutes. And I hope that the workflow that I've shown you is applicable in your work.